Hey, it's Glenn Wokenfeld, and this is This Week in AP Biology for Teachers. We're going to talk about transcription and translation and how you can use the interactive tutorials on sciencemusicvideos.com or learnbiology.com to make your students' achievements soar. You'll see that if you stop using your textbook or use it just a little bit and use the interactive tutorials on our websites instead, your students will learn so much that you'll be delighted. It'll feel great about for you in terms of what you're doing. They'll love it, and let's just see how that's going to work. So imagine what it would be like if your students came to you and they could tell you about the details of transcription, about the role of RNA polymerase, about how free nucleotides were taken out of the nucleus and then combined to make the various RNAs that are involved in the processes of transcription and translation. Or if your students came to you after doing a homework assignments and they were completely able to explain how protein synthesis worked. They knew the details of the genetic code, and they could use a genetic code dictionary like this one to explain the details of protein synthesis. That would feel pretty great. That's what learnbiology.com, learn-biology.com, don't forget the dash, and sciencemusicvideos.com are all about. So let's start by talking about this from sort of evolutionary perspective. We'll talk about the four big ideas that are part of the AP Biology curriculum. So in terms of evolution, transcription and translation are about as fundamental to life as you can get. And in fact, these are processes that are found in all living things. And, if you think about what happens during natural selection, you wind up having changes in DNA, and those changes get transcribed into the various RNAs, and those get translated into proteins. And you can sort of think about it as you have genotype up here and you have phenotype down here. And what's phenotype about? Well, the phenotype is about creating systems that can recreate the genotype. So you have, for example, in the case of the SARS-CoV-2 virus that's causing the COVID-19 pandemic, you have a virus whose genome is kind of randomly mutating, recombining over this kind of adaptive landscape. And what does that lead to? And of course, the genome here is an RNA genome, not a DNA genome. But what that winds up with is producing a spike protein, and that spike protein is capable of somehow adapting to bind with the ACE protein that's found on the epithelial surface of our respiratory protein. And that made possible the spillover that let the virus jump from bats, who had been almost certainly its previous host, into humans. And 60 million cases later worldwide, and going on 1.5 million deaths at the time of the making of this video. It's hard to not wonder at this adaptation and how effective it was in this case in terms of letting this virus reproduce the way that it did. It's an amazing example of natural selection. We can talk about other kinds of key themes of biology that are related to transcription and translation. So, for example, information flow. One of the key themes of AP Biology, well, this is all about information flow. We have DNA triplets that are being transcribed into RNA codons, which are then being translated into amino acids. It's all about information flow, and that's of course an inward to outward information flow, but you can connect this to cell communication that we just covered a couple of weeks ago. And remember that there are hormones that come into the cell: estrogen, testosterone, growth hormone, and those bind with cytoplasmic receptors that then act as transcription factors that turn on genes and affect the proteins that the cell will make. So there's a lot of connection there. You can talk about energy. This is a highly, highly endergonic process. You're taking free nucleotides, creating them into organized strands of RNA, and then you're taking free amino acids and organizing them into highly structured proteins. That's highly endergonic. That doesn't happen for free, and that requires either ATP to drive ribosomes over here, or remember that the free nucleotides in here, they come in like ATP. They're triply phosphorylated. You've got your G ATP and your UTP, and those phosphates are providing the energy that's enabling RNA polymerase to drive the reaction of creating a polymer from a monomer. And of course, systems, here's the system at work right here. But what's cool is that this system can be extremely simplified, and you can run in vitro protein synthesis, and that's old technology. This has been done since the 1960s, and that's how Marshall Nirenberg figured out how he could create artificial mRNA that just had, I believe, A in it. I might be getting that wrong. But in any case, he was able to create a single a polypeptide that had only one of the amino acids. I think it's phenylalanine. I might be getting that wrong too. But that was the first step in terms of figuring out the genetic code. So we've got everything that's here. And it's really important as teachers that we make these connections for our students. So let's go on a little bit more and let's talk about what the College Board is asking us to be teaching about in this unit. So just so that you know, I have available on my website this document, and I took the College Board's course and exam description, and I just boiled it down into a Google Doc, and then you can jump around and you can go here to Unit 6. And last week or 2 weeks ago, we talked about replication and DNA structure, and now we're talking about transcription, which starts over here, and translation. And I took these objectives from the College Board and I boiled it down into this. And again, just so that you know, in my curricular sequence, for a variety of reasons, including what I think are good pedagogical reasons, we're doing topic 6 before topic 5. We'll come back in 2021 and we'll work on topic 5, heredity. So in terms of what the College Board is looking for you to teach about in terms of transcription, there's the general flow of information that your students should have mastery over. The idea that DNA makes RNA, makes protein, that involves several types of RNA, messenger RNA, mRNA, rRNA, and then tRNA. And then the process by which RNA polymerase actually transcribes RNA from a DNA template, and then how that mRNA transcript that emerges in the nucleus is modified in eukaryotes before translation occurs. Though I want to emphasize that I don't cover that in the tutorials that I'm going to show you. I cover it later in a module about eukaryotic gene regulation. And now let's look at translation. Translation, the College Board is asking you to talk about how ribosomes actually work, how they're translating mRNA into sequences of protein, and there are key terms like codon and anticodon, the roles of the mRNA, tRNA, and ribosomes, which are partially made of rRNA, the start and the stop codon, and the whole shebang involved in this. So you really have to go into some detail in terms of teaching this according to the College Board's learning objectives. Note that the College Board is also asking you to talk about how retroviruses reverse this flow, but I don't cover this here. I have a whole module about viruses, and you don't want to skip that in 2020, 2021, when a pandemic caused by a virus is changing everybody's life. So we'll again hit that either next week or it'll be the first thing that we'll do after winter break. So let's go on to Science Music Videos and talk about how we cover this material. So you'd start over here at our main menu. You'd hit the AP College Bio menu. That takes us to our main menu, which covers the entire AP Biology curriculum, though truth be told, I'm still working on the last one. I'm going to finish it. This one down here, ecosystem disruption. It's going to happen over winter break. I'm really hoping I get there. And here we are. So we're in this topics 6.3 and 6.4 from gene to protein. So here's what we do on Science Music Videos. Note that there's a student learning guide that goes along with this. So you can open it right here. And here it goes. It's a Google Doc. And, as the name says, what it does is it guides your students through everything that they'll need to know as they do these tutorials, and it'll really work them towards mastery. So use the student learning guide and you'll have even better results than just using the tutorials. So let's go back here, and we start with a tutorial about transcription. And what I always like to do is connect this to basic processes in cells. So interactions like this, where kids are reminding themselves that like this is transcription of DNA to RNA. Oh, I'm not logged in. Oh, that's okay. Well, you can sort of see how that works. I'll log in later. Maybe I'll log in right now. Okay, so here I'll log in. And there we go. So I'm logging in as a student, and this gives us that fantastic preview. So this is a premium feature on Science Music Videos. I totally encourage you to get this, because if you do that, you can go like this, and then you can see, well, in this case, all the answers to this. And oh, this is a one-question quiz. All right, so now back to work over here. So this gives you the big picture, and then what I'll do is I'll teach about the three basic kinds of RNA: ribosomal RNA, tRNA, and that'll be followed by a quiz. This part of the tutorial will talk about the differences between DNA, deoxyribonucleotides, and RNA nucleotides, which have this hydroxyl group over here instead of a hydrogen atom. And it'll teach you about the base pairing rules. So like, for example, over here you have to drag in the matching nucleotide and RNA. If you have an RNA, so RNA to RNA, so A would go to U. Let's see if I got that right. Fantastic. And let's see what happens. U goes to A. So, this kind of thing. That's just building mastery about base pairing rules in RNA. And then an interactive reading about the process of transcription, so your students can, completely understand how transcription works. And that culminates in some flashcards and also a quiz. So I think that gives you enough of a sense of that. The next tutorial will walk your students through the genetic code and how that works. And I start with a couple of coding exercises, like the basic idea of how it is that just 4 letters can spell out a 20 amino acid alphabet. And I do that through this coding exercise, where I sort of have a coded message here that I wrote out in these series of nucleotides. This activity goes back to Paul Sherman, who was one of my mentor teachers when I taught at Wesleyan High School in Oregon. And Paul's passed away many years ago, but whenever I see this activity, I'm grateful to him and the mentorship that he gave me. So this teaches you about the basic idea, and then I actually walk students through how they can use a genetic code dictionary. Because students will see both, I provide them with both circular dictionaries and with more traditional tabular dictionary that you probably grew up with when you were first learning how to decode. And then there's a bunch of decoding exercises. And by the time you're done, students will really understand how they can take messages in DNA and, messages in RNA, code them into protein, and actually even take messages in DNA, work backwards into RNA, and then work backwards into protein. So mastery of the genetic code will come about through that tutorial, and then we go into protein synthesis. And again, I'm constantly showing students what the big picture is, and your students should be able to talk through a diagram like this. That's always one of the things that I'm aiming for as a teacher, like, can my students actually explain what it is they're learning? And if they can, that's really setting themselves up for success on FRQs and other activities like that. So talking about a little bit of review of protein structure, amino acids, primary structure, what peptide bonds are, and then showing how a review quiz about proteins, and then showing about how the actual process works, about how you have tRNAs that are charged with amino acids, how they have an anticodon, that anticodon matches with a codon over here, and the whole process. I have I have a lot of songs, as you know, and really one of my favorite ones is this one about protein synthesis. And it really explains the whole process with some really great animations, and I totally encourage you to play this song. I'm going to hold off from doing that right now. I'll just leave it for you. You can find it here. I'll also show you where it is in another part of my website. So the last tutorial in this sequence is not actually part of the official College Board learning objectives, but I feel like it's so incredibly important for students to understand why certain proteins are made at the rough ER, whereas others are just made in free-floating ribosomes. That's sort of essential to how the endomembrane system functions, and it seems like it's an important thing not to skip. And so this tutorial will talk them through how that process actually works. It talks about how all ribosomes start as free ribosomes. As they're translating, the first couple of amino acids can serve as a signal, and that signal gets bound by a signal recognition particle, which docks at a receptor that's in the rough ER, and then the resulting protein is threaded through. This is a diagram from bioninja.org, fantastic website. He shares stuff with me more so than it flowing the other direction, but I can't really recommend BioNinja enough. It's fantastic. So this will walk you through the whole process, and it'll end with a quiz. So that's pretty much it in terms of the stuff on Science Music Videos or learn-biology.com that'll guide your students to mastery about transcription and translation. And to end, let's just go back here. And there's a couple of additional things. For one thing, Flynn has two really good Pogles about transcription and translation. I think I've got one up here. Yeah, there we are. So Flynn, Scientific, they have these great worksheets called Pogles. And actually, if you do this Pogle about transcription, it has these models. Your students answer questions about them. If you're completely online, you might want to modify them a little bit so it's a little bit easier for your students to fill in the answers. But they also do talk about mRNA processing right here. So if you want to hit that objective right here, you can do it just through this Flynn Pogle. And there's an equally good one about translation. So that's highly recommended. And another thing I wanted to show you is this. So where would this be? If you went to For Teachers, and then you went down here, Teacher Handouts, and then you went here, Protein Synthesis and Genetic Code Worksheet. So here it is. You can download this as a PDF. This has the same kind of decoding exercise that I talked about earlier, but in paper form. This has another one that's really good in terms of your students understanding that you have to do two transformations to get from information in DNA, that's kind of what these symbols represent, to information in RNA, which is what these numbers represent, and then ultimately you'll take that all the way to letters. Some information about proteins, transcription over here, and then some really good exercises about translation. So, go ahead, download this, use it, modify it. It's pretty good stuff. I'm not using this right now, at least not directly, because I'm not lecturing to my students. And the reason why I'm not lecturing to my students is because I'm having them learn the stuff through the tutorials on sciencemusicvideos.com. I've talked about the MICE method, where what I'm doing is I'm motivating my students, giving them a quick introduction. Then I'm having them do interaction on the website, on the tutorials. And then what I'm doing in class is using time in breakout rooms. I'm completely on Zoom to have the students discuss the material. I'm quizzing the material using the timed quizzes that I've started developing. You can read more about that in my newsletter. And having them do other activities. There's not, like the Pogle that I just showed you. So that is pretty much it for this week. I want to thank you for your time. Again, if there's one word to remember it's interaction. Get your students to interact with the material, and their achievement is really going to soar. And I just totally encourage you to do that, both in your direct teaching with your students and in terms of the assignments that you're giving them. And that's why I'm using Science Music Videos. I wrote it for my students, and I want to encourage you to use it with your students. And Science Music Videos, it's interactive learning. You're going to be astonished at the A's your students will be earning. Thank you so much. I'll see you next week. Take it easy. Shoot me an email if you have any questions. There's information below, and it'll show you how to get to my website so we can continue this conversation. Have a great week, and again, thank you so much.